let's continue with this last talk before the coffee break, not last talk for today. And I'd like to introduce you Anna Levina from University of Tübingen. It is a very interesting topic, not my field, but that's why I'm very excited about the topic as well. Thanks, Anna.
Thank you for the introduction, and huge thank you for inviting me here. It was very inspiring meeting so far, mixing so many different fields, and I would return back to neuroscience part. So my talk would be about timescales, but I would mainly mean neuroscience timescales in neurons and behavior. Why do we think that it is so important? We already seen a little bit glimpse of maybe it is good to make neurons, artificial neurons, slower, and maybe time is something that is very important. Let's look at it again. And the magic. Where is the magic? OK. How do I get it to work? OK, now it works. So when we think about time in natural tasks, like typically, if it is at your home and you see this picture, you know you need to act very fast. You would just perceive the image and act. If you plan what to take with you for going out, whether to take an umbrella or not, unless you are now in southern Germany, you might contemplate taking an umbrella or not, and it might be wrong or a correct decision, but both the time or the integration of information you could use for making this decision and the consequences of this decision would be relatively short in time, like maybe one day or a few hours. And sometimes there are decisions that really require very long integration of information and have consequences for a very large part of your life, like deciding what to study or what to do with a big chunk of your life. The crazy thing about all these three tasks that at least for humans and for animals, we do them all in the same system, in our brain. So it seems that the brain and our potential, at some point, generalized system would need to be able to decide flexibly over which timescales to integrate information for which decisions, and also act at different timescales. And additionally, if we think about biological or physical systems, they have natural constraints. The biological processes can't run infinitely fast, and they have hard time staying the same for infinitely long time. So we don't have very fast and very slow timescales. And today I would be talking about three different facets of looking at timescales in different systems. And we would start with looking at the timescales in the data. This would be a data from a visual area of a monkey. And our question would be, how do the timescales change with attention? Then we would try to interpret what we learned from the data and look at the artificial systems that actually make a task and can flexibly change timescales to solve the task better. And we finish by trying to understand what are the general principles, what do we need to include if we want to emulate a single neuron, and how making this cleverly can help us to make good computations also on larger scales. So let's dive in. It would be a more like detailed talk on the stories. So we'll start with attention and timescale in the brain. So for neuroscience, for a long time, there was already a hypothesis that because we have these different brain areas that involved in different ways in the computation, there should be potentially different timescales that are dominating activity in different brain areas. In a sensory system that just immediately sees or hears something and reports, it should be short timescales, and the higher we go into the cortical hierarchies, this would be slower and slower timescales. Indeed, this is what was noticed. Here, we go in a hierarchy from areas that are closer to the vision, to the areas that are closer to the complex computations. And the higher up we go in this hierarchy, the slower are timescales. The same was found with the different ways of getting information from the brain. On one side, looking at the spiking activity, looking at the ECOG, which is like a more precise EEG, or at very slow fMRI timescales. Everywhere it seems to have some kind of this trend of having a slower timescales where the more sophisticated computations happening. Additionally, what people found by looking in more details in timescales during the computation is that neurons that are involved in doing some tasks might have different timescales depending on which part of the task they are involved in. And after performing a memory task, the timescales in multiple brain areas could become longer. So there is the flexibility of timescales and flexibility of using different timescales for different parts of computation. So my main question in this study would be whether we could see the localized timescales in a small neural population, whether they are changing with attention in this case. And we would try to find out whether we can come up with a good mechanistic explanation of how these timescales can reasonably come about. And this is a study primarily done by my very bright PhD student, Roxana Zerati, in collaboration with a group of Tatiana Engel, now in Princeton, and her postdoc, Yang Shi. So first of all, let's pin down what do we think about when we say timescale. And the mathematical way of thinking about timescales is we look at the autocorrelation function. So here we take a signal, we shift it by a predefined lag, and look at the covariance between a signal and its shifted version normalized by the variance. This will generate an autocorrelation function. And this function typically looks like this. It is normally an exponential decay, which we could even nicer visualize if we plot it in logarithmic linear scale. This is what I will be doing a lot in this talk. And then the slope of this exponential would be my timescale. Or if I would fit it here, this would be this tau. So now let's come to the data. And the data, it was recorded from the area V4 of macaque monkey doing an attention task. So here, what we need to know, what we've seen already in Panayota's talk, is that the neurons have this receptive field, so they're looking only at a part of the total visual field. And here, I marked the receptive field of the neurons the experimentalists were recording from. In this task, a monkey would need to define whether the GABA patch changed or not changed in one of these 4 locations. And before starting identifying the change, it would be given an attentional cue. OK, it should be here. Then it would need to make a saccade opposite to the direction where it's seen a change. So if attention was given over here, then this and this would be somewhat related to attention, but these 2 directions would not be related to attention. So if we record from neurons that are kind of concentrating on this part of the visual field, this would be an attention away condition. So let's have a look how this autocorrelation function look like. And this is the same representation in logarithmic linear scale. After looking at tons of this, what I see here is that there is a fast decay in the beginning and slower decay towards the end. We'll see where this is coming from. So my first hypothesis could be formulated as that there are maybe multiple timescales, potentially 2 timescales, that are good to describe this data, and 1 timescale would not be enough. And if I would look at what is happening if we record from the attended direction, we would get this autocorrelation function that looks very similar in the beginning, but very different towards the larger lags. So I would hypothesize that the slow timescales would be increased because of the attention. To check this hypothesis, the first thing we would need is to have some statistical way of assessing these timescales and actually being able to formulate and decide on the hypothesis about them. And there is a small problem with it. Namely, the neuroscience data is typically organized in short trials. And if we would look at the short trials from the absolutely perfect Ornstein-Uhlenbeck process that has the timescale, I know I put it in. If I make the trials as short as 1 second, which is a typical for monkey data, my direct evaluation of timescale would be very different from the one that was put in, and the original timescale was 80 milliseconds. And we had to go to develop the Bayesian method of estimating timescales for this purpose. So if you are interested and have short trial data, look it up. But now let's just assume that we have the method that allows for very good Bayesian evaluation of timescales. And now we have data from a single session. So we can fit it with the two models. One model that has a single timescale and another model that has two timescales. It looks like one of them is better than the other, but as always, we would need to do the strict hypothesis testing that accounts for the fact that one of them has more parameters. And indeed, if we do the Bayesian model comparison, what we find is that in almost all the sessions, the model with two timescales is the one that fits the data best. Sometimes there are inconclusive results, which typically means that the firing rate was too low, and we did not really have enough of the data for evaluating autocorrelation function. And in some few cases, there are single timescales winning. But at least we see that in most cases, most of the data for 3 monkeys that we had data from is well fitted by two timescale model. And now we can go to this attention and non-attention task version. And we see that now there is a data fitted by the optimal two timescales model. And if we look at the short timescale, the tau1 is a short timescale in the beginning, there's around 3 milliseconds, they are very much overlapping. The posterior distributions, because it is a Bayesian method, we have a posterior distribution of all possible timescales that could explain our data. They are overlapping almost completely, and for the long timescales that related to integration of information over longer times, there is a very clear difference. With attention, they tend to be much longer than without. And if we pull the data from all the sessions we had, the trend remains that it is strongly different for the attention towards, and it is much slower for attention towards the receptive fields than from attention away. And what was particularly exciting for us that when we looked at the data and at the behavioral part of the data, what we've seen that if the monkey was attending towards the stimulus, and we recorded from the receptive field of the neurons that are in the attention focus, then the longer the time scale, the shorter would be the reaction time. So if the monkey was integrating information over a longer time, it would be faster to react. And in the attention away, there is no connection between the time scales and the reaction times. So here we establish that there is something happening. Attention is something very fleeting, but still it is changing the time scales of local populations. And what we tend to do now is to check which models would be compatible with our observations. It's the simplest way to introduce two timescales into the activity is to say, I should have just two processes on these two different timescales. For example, I could have an individual neuron being relatively fast and synapses being relatively slow. And this is totally valid mechanism of generating diversity of timescales we could do for any two timescales and match the data perfectly. The option that we were more excited about was an option to take neurons that would be relatively fast, that would individually have this fast timescale, and to generate the slow timescale by recurrent interactions within the network. So all neurons are the same. There is no other timescales in the model, but there are spatial connectivity that generates a strong recurrence. And we found analytically that we can, by using a simplified spatial network, produce any slow timescale this way and generate the 2 timescale processor, which pose a problem. We have now 2 methods that generate the same pair of timescales. So we need somehow to select between them. And the perfect option for us was to look at the cross correlations in space. Sure enough, if we take a random connectivity and to make a very distilled method, we took this 2 timescale version with random connectivity, there is nothing happening in the spatial correlations. However, if we take this spatially distributed network, we would have on one side a decay of cross correlations if we are going further in space. But the other thing that we observe here and that we could show analytically, that the further away the neurons are, the further away goes downwards to the lighter shades, the further away the neurons are, the slower timescales that are dominating the activity. So on the long distances, neurons are communicating primarily over slower timescales. So that's all good. Now we need to find some features in the data that would allow us to select between these 2. And to our luck, some of the electrodes were inserted not very perpendicular to the cortex surface, so it was possible to say that they are spatially a bit different by looking at the receptive fields of the neurons estimated from presenting them additional stimuli. And this way, it was not very large distances because it is still a single lamina probe, but we could separate the short receptive field distances from the long receptive field distances. And you can maybe appreciate that there is an ordering that is similar to the ordering we have, even though the shape has some deviations. And if we would just measure the time of this, the time scale of this slow cross correlations, we would see that at the long distances they are slower. And we could show in analytics that this should be what's happening for the spatially structured network. So this way, we decided that the spatially connected model is a better match to the data. So to summarize this first part, what we've seen is that there is a strong support that local activity has multiple timescales, predominantly 2, and that the slow timescales are modulated by activity and have some behavioral relevance. And that the model that can best reproduce this 2 timescales structure is a model with a spatial connectivity, where the timescales are the slow timescale is generated by the recurrence. So now let's turn to the artificial neurons to be able to study which of the 2 options would be preferable for solving tasks. So we've seen that potentially these 2 could be good. In the biological data, this one should be better, but why this should be the same way for solving artificial tasks? So we would ask what one is better if we need to solve a task that requires memory. And this is a project that was mainly done by Sina Haji Abdullahi together with Roxana and Manas Yanakakis, who has a non-related to it poster tomorrow. So what we took a discrete version of a model that very much resembles the leaky integrated fire neuron, just that every neuron is allowed to have its own timescale. And there were already multiple studies showing that it might be very good to allow for this variety of timescales to be computationally efficient. So these are our timescales that are forming the same principle as the leak constant of the neuron. And these all blue parameters are trained through backpropagation through time. So now, what are the timescales that we would care about? Like we have this one that we train, but in general, the network does not care explicitly about this timescale. It cares about the timescale of the output of every neuron. And we can measure it. We can just record the activity of the neuron, generate again the autocorrelation function, measure the timescale from it. And we would call this an effective timescale because it is not the parameter that we put in. It is a combination. It's influenced by the parameter of the neuron, but it's also influenced by the recurrency. And what we know from our analytic study is that if the connections are very weak or very random, or both, then the effective timescale would be very similar to the individual timescales of the neurons. But if they're strong, it would be larger. So now we can get a task. And we want it as simple as possible task where we would still need a memory and we would need a progressive amount of memory as the task getting harder and harder. And for this, we took the n-parity task, where a network needs to compute the binary sum of a previous n inputs and decide whether, so in the end, it would be either 0 if it is even or 1 if it is odd. And it would need to report the output at every moment of time. So it would need to always compute it backwards. For very short n's, it was already explored and it was seen that something very similar to our effective timescales need to be longer and longer the larger n's we consider. But we want to go for really large n's. And the first thing we tried failed miserably. So this is how long we need to train if we just want to solve for n equal 5, which is very small. For n equal 10, we still can solve it in a reasonable computational time, but if it is 15 or 20, we fail. So we need something to teach these networks better. And one of the options to teach networks better on progressively hard tasks is to use some curriculum. And we came up with two different curricular options. One would always try to just learn the next task. It would be kind of a pre-trained on all the previous tasks. Like say we start with n equal 2, then we take the recurrent network and continue training from this point for n equals 3, 4, and so on. The only problem we have there is that the network that is trained, for example, for n equal 20 cannot solve anymore n equal 10, which is sad, but maybe it is fine for us. However, if we want to mitigate this catastrophic forgetting, we might instead train the recurrent network together with the multiple readouts to be able to still continue solving all the tasks. So we would train for n equal 2, and then together with n equal 2, we would start training n equals 3 after the 2 is completed, and then 4, and so on. So the last network would be able to solve all the tasks with smaller n's. So now we have these two different options to train them. Let's have a look how they perform. And the first thing we see is that if we train for the single task, we can solve much larger n's. So it is better than not having a curriculum at all. But if we train to solve multiple tasks simultaneously, which might feel like a harder task, it is actually a very nice bias to be able to solve a larger n's. And if we take the network that already solves the task and start perturbing it, the ones that were trained on multiple tasks are more stable to the perturbations. So now let's come to things that we actually wanted to do. We wanted to measure the timescales and want to look which timescales individual neurons preferred. And what we were interested to find is that there are different dynamics of individual timescales for these two different options. If we train in the single head only one task at a time, from some point on, we try to generate the long timescales apparently by increasing the timescale of individual neurons. However, when we learn for multiple tasks simultaneously, it seems like the timescale all the time decays. To check it in a bit more precise way, we just fix the timescale at the shortest reasonable time, and the shortest reasonable time is amount of time every stimulus is presented. So it is kind of a time scale of stimulus presentation. And if we take this multi-head network, whether we fix the time scale or we train it, there is no difference. And the only problem starting if we fix the time scale to be too slow, and then the network starts to be able to train only for smaller sizes of the tasks. However, if we take the single-head network and we prohibited learning the timescale, it's unable to solve the task at all. And now we look at the effective timescale. So the timescale, that is the timescale of the output of the neurons. And what was interesting for us to see is that they behave relatively similar in both of this scenario. So they all growing within relatively close, following each other for a long period of time. However, because we see that after some time the single-head networks is learning on one task at a time starts to increase the time scale, we could assume that in this driving the increase in effective time scale, there is an impact of the individual neuron time scale. For the multiple tasks at a time version, we know that we can just fix this time scale at the lowest reasonable level, and it would be capable of solving the task. So the all lifting for changing the effective time scale would be delegated to the recurrent connectivity. And to summarize this part, what we've seen is that in a single task learning in single-head curriculum, the timescales rely to some extent on the single neuron properties, whereas in the multi-head curriculum, the timescales only rely at the network interactions. And it seems that in these two networks that solve very similar tasks, it is much better to rely on interactions instead of using the single neuron property, which seems to be compatible with what we've seen for the monkey, where it's the long timescales were generated by the recurrent interactions. And now, briefly, let me go to the last topic of looking at the individual neuron. And as computational neuroscientist, I'm used to the neuron that is a single number with few additional numbers that would mark the connections. However, neurons are much more complex. We've also seen it in the talk of Jota before. They have a complicated morphology, and they do complex dendritic computations. So the task here would be to look at the biophysically motivated, very complex model of a single pyramidal layer 4 neuron that has around 1,000 inputs in this model. So it is a model with very many compartments. Every compartment has multiple differential equations. It takes forever to simulate. But we did not, because there was already a very cool study trying to match the input-output of this biophysical model that is as good as we can do now to characterize all the input, all the outputs of the single neuron by using a temporal convolution network. And they were quite successful. So they could reproduce the output of the neuron both in terms of spikes and in terms of changing of voltages. And Aaron, who is here and who can talk with you about the details of what I'm talking about at the poster tomorrow, proposed a different approach. So instead of having this very rigid temporal convolution network, let's look at the recurrent module that would have multiple memory units with different timescales. In a biophysical way, we could think about them as implementing a different set of computations at different timescales. The input comes through trainable nonlinearity, integrates together these memory units, and this nonlinearity generates on one side update for the memory, and on the other side an output in forms of the predicted voltages or spikes or both, depending on what we want. More formally, we would generate this nonlinearity through multi-layer perceptron, and we would also, for biological realism, add a synaptic filter because synapses in real neurons are not instantaneous. And with this model, which we called expressive leaky memory neuron, we could very closely reproduce the input-output relationship, but this is not as surprising because there was already a model that was reproducing it quite closely. What we would look at is how many parameters we need for that. And here, we match this input-output relationship. First, this dark-colored dot is the original temporal convolution network. We can also, using our recurrent idea, because all the differential equations in a sense are like a recurrency in itself, we could just use LSTM. And we could also get a very close fit of the voltage and good prediction of spikes with LSTM. But we would need quite some parameters. And both blue models are our suggestions with this trainable, continuously changing memory units. And now I show you what is the difference between them. The light blue one uses slightly the morphology of the neuron. It takes the input and cuts it in multiple chunks, which it would sum and then get through nonlinearity. However, every input would be a part of multiple of these chunks. This way, allowing also nonlinear summations of the inputs within a dendrite. If we would take an individual chunk for every dendrite, we would have much lower performance. But this way, we would reduce the dimensionality of our multi-layer perceptron, which is the largest parameter source in our model. And we would get, with slightly more than 10,000 parameters, already a very good fit, which is now 3 orders of magnitude less than the previous model. And we need to assume that, or need to remember that there are thousands of parameters that are just synaptic weights of every input. So 10,000 is not much more than we have in the input space. And now we could use this model for learning something about biological neurons. I just show you a simple example how we could imply it. We could change the hyperparameters of the model and see at which point it breaks and stops following the real neurons. For example, here what we did, we changed the number of memory units. We did not allow more than 1. And then almost none of them work, except if we would want not to match the neuron, but to match a simple leaky integrated fine neuron that actually has only a single time scale. And then as we go with the more and more complex neural models, here we come to this biophysical model of the cortical neuron, and before we have the Hodgkin-Huxley model that is already have also quite some number of differential equations, we would need more and more memory units to match it. And additionally, what we could do, we could say, OK, forget about biological constraints, use as many memory units as you want, and train on a really hard task. A really hard task to highlight is here this Pathfinder X. This is a task where a network needs to determine whether there is a continuous path between the dots. Like here there is one, and here there are none, even though there are passes from every neuron. And to make it harder, this is not fed like this to the network, but is fed pixel by pixel. So it is very hard temporal dependency task with 128 by 128 pixels. And our network is not better than the best, but it solves it where the vanilla transformers can't. So altogether, our LM neuron model could nicely match the input-output relationship. It produced something that we could interpret afterwards to design the neural models. And it might be a powerful way of solving long memory tasks. So now, to just sum up everything, what we've seen that in the data, there seems to be multiple timescales, even if we would look at the small part of the visual cortex, and this timescale seems to be modulated to solve the tasks, in this case, visual attention task. That it seems like also in artificial networks, there might be a benefits of changing the timescales by virtue of recurrent connectivity and not by virtue of individual parameters. And that using this type of recurrent structure with multiple timescales could be a very good way of fitting very precise and detailed biophysical models, and this way representing the real biological neurons. So with this, I want to thank my lab and collaborators in theory and experiments, my funding sources, and I would like to get some questions.
Thank you very much. Yes, again, just the same game as before. If you have a question, just please feel free to use the microphones there. And we have already one question, please.
Nice talk, Anna. Great job. So I didn't quite, maybe you said this, but I missed it. It's evident that attention increases the time scale. Mm-hmm. Do you know in your networks how you would model attention in order to capture that aspect of the data?
Mm-hmm. So in the most simple way, we would just make the connection stronger, a little bit. The good part is that we would not need much. We did try matching it directly with this model, and it's a very tiny change. However, our model is very linear. If we introduce a strong nonlinearity, actually the same way the changing of the connection strengths can be matched by a very tiny additional input that would, for example, be able to come from neuromodulatory system. So we can match it more precisely, but if we stick with this quite linear model, this would be by increasing a connection strength by something like third order after comma strength, so it's not much.
Thank you. I see there's another question, please.
Hey. Thank you very much for the very nice talk. I have a question more on the experimental side. Like, those data that you showed were the actual correct examples, like where the monkey was able to actually do the task or not?
Yes. So we analyzed primarily the correct one, but I think, so this is not very hard task. We can't, so we also analyze the non-correct ones.
Right.
There is no very clear correlation with correctness of almost any observables there.
So you see the same on both cases?
Yeah, but we can't, so there is no predictor from the time scale to whether this would be correct or not. So it's not, we did not see anything significant. We could somehow do only interpretive dance.
Yeah, because I was just wondering, like, if this increase in the autocorrelation could just be explained by the fact that there's a correct trial, so you have more bottom-up input, and that would just create some recurrent activity, and you would just get longer autocorrelation function, but there's not really an attention mechanism going on.
So it does not feel, I understand you, it does not feel to be this way. It's more consistent with the idea that there is some kind of potentially like slow fluctuation of the internal state that would modulate both how fast the monkey reacts and what are the time scales.
Yeah, and maybe can I just ask a follow-up? Like, actually, it's not a follow-up, but it's on the other side. It's more on the neuromorphic implementation of that. Like, would you suggest to have more detailed models of single neurons in a neuromorphic chip, for example, that has multiple timescales, or you would just suggest that we should look at different type of architecture that will create in the network level longer timescales?
So what I see, and I'm using here a network that is very compatible with the neuromorphic chip because it is actually a nearest neighbor interaction in some radius. That this is sufficient to generate long timescales without having a strong kind of individual neurons change and variety. Whether physically this is the best solution, I don't know, but potentially this could be a simple enough solution that might work.
All right, thank you so much.
Thanks, everybody. Well, if there are no more questions, please thank Anna again.